I know you're so excited to continue learning in chemistry, and that's exactly what we're about to do. In our last part of our section, we learned about atoms and molecules, and you should know the three subunits found in an atom and the difference between molecules and compounds. You should be able to, if I get told you, let's say a solution has a pH of 3, you should know if that means it's an acid base or if it's neutral and why. And probably a lot of other things, right? Today we're going to talk about carbon and its importance in life. Carbon is the fundamental building atom for what we call organic molecules. Now, don't think of organic like what you think of when you go to the grocery store. Ask students all the time, what does organic mean? And they think it means naturally occurring or common or abundant. And that is not the definition of organic in chemistry. Organic molecules are molecules that have a framework of carbon, and with that carbon attached is hydrogen. So we're going to put some atoms together to make up some pretty complex molecules in this section of the chapter. So what are inorganic molecules? Well, inorganic molecules aren't made of both carbon and hydrogen. For example, salt, water, CO2, things of that nature, larger molecules. Those are inorganic molecules. But if you have a molecule made of just carbon and hydrogen, we're going to call that organic. And the simplest one is CH4. Now, there are some major complex ones like I mentioned. For example, antibodies molecules are huge and have specific shapes, and we'll see that in a little bit. So carbon is ideal because it is that atom is abundant, that atom is everywhere, and because it has 4 valence electrons in its outer orbital, it needs to share 4 other electrons with other atoms. And so how's it going to do that? Remember, when you share valence electrons, that forms what type of bond? That's going to be a test question, so you should know that it's going to make covalent bonds. So carbon's going to make 4 covalent bonds all the time. And a lot of times it's going to be to hydrogen, as you can see in these pictures. And what is also neat, carbon is small, and it can make all these different shapes. It can make rings, it can make linear shapes or branch shapes. It can bond to not only a number of other atoms, but carbon can also bond to itself. And so all of these things make carbon the basis of all these organic molecules. It makes it easy to make a bunch of different structures. Now, functional groups are these little groups of atoms that you can attach to carbons, and it totally changes the structure of that molecule and therefore changes the function. As you've learned in previous science classes, structure determines function, and when you change the structure, you change the function. For example, putting an OH on a group of carbons makes that an alcohol, and we'll see that fatty acids often have carboxyl groups, and a carboxyl group is a carbon double bonded with an oxygen and single bonded to an oxygen with a hydrogen attached to that oxygen. Now, this R over here is a variable group. And what that means is this is just a group of carbons or another molecule you're attaching these to. Lipids are going to have esters. We'll hear about esters bonds. Sulfhydryl groups are pretty interesting. They're not really common. They're only in one specific amino acid, that's cysteine. If your protein has a number of cysteine amino acids in them, those sulfurs will actually be attracted to one another, creating a disulfide bond. That's just a fancy term for a double covalent bond between sulfurs. And really what you want to focus on is the phosphate groups, because we're going to see a phosphate group in DNA, we're going to see a phosphate group in RNA, and ATP has 3 phosphate groups. All right. Sorry about that. So macromolecules are large molecules, and we have 4 main types of macromolecules, and this is where you're going to want to focus for this part of the chapter. They are carbohydrates, lipids, proteins, and nucleic acids, and these were also in Fundamentals of Biology as well as Anatomy 1. So carbohydrates, they're subdivided into types based on their structure. And so we have monosaccharides. Mono means one, saccharide means sugar. So that's a simple sugar. We have disaccharides, double sugars. And we have polysaccharides, which is a long chain of sugars. So monosaccharides like glucose and fructose are going to be involved in metabolic reactions. So what we're saying is these two are examples of monosaccharides that can come together to build disaccharides and even polysaccharides. Now, the function of carbohydrates, generally speaking, is they are your number one source of energy. So if you eat a diet heavy in carbs, and then your body's going to burn that first for energy, and anything that you don't burn or use for energy will be stored as fat. Okay, so carbohydrates help with your primary source of energy. They also are going to be found in cell walls, and so we'll talk about those later on, and food storage. So our disaccharides are maltose, lactose, and sucrose, and I bet you've heard of lactose before because some people are lactose intolerant. So lactose isn't milk itself, it's the sugar found in milk. But a big clue that it is the sugar in milk. Lact means milk and ose means sugar. Notice glucose, fructose, maltose, lactose, sucrose, even cellulose, that suffix ose is telling you that it is a carbohydrate. Maltose is what you make when you break down starch, which is one of my favorite things to eat. I love a McDonald's French fry and all its goodness. And then sucrose is very common. It's what you put in your sweet tea or your Kool-Aid. It is your table sugar, and it's made of 1 glucose and fructose. Lactose is glucose and galactose, and then 2 glucoses together is maltose. Chains of these monosaccharides are, as I mentioned, polysaccharides, and this is going to be starch, like I said, my favorite. And then cellulose, which is in plant cell walls, it is what we might call fiber. We are not able to digest it well, and it will help you go if you know what I mean. Glycogen is what we store sugar as in our liver, in our muscle. And so we can temporarily store some sugar for when we're going to need it. I think about an athlete carb loading before a athletic event, and they're going to have the complex carbs. Your body's going to break those down and then store them in the muscle temporarily. Lipids, their primary function, is your second source of energy. So if your diet is low in carbohydrates, then what is your body going to use for energy? The next thing it goes to is lipids. So lipids provide some energy. It also is super important because it makes up cell membranes. There's some waxes that make up cell walls. And lipids also make up steroids. Steroids can be cholesterol based or structures, and we'll see these in the cell membrane. We'll also see these in animal hormones. Proteins have a ton of functions, and they are probably the most abundant macromolecule in the body. They are all kinds of shapes, and it's just wild. A long time ago, we thought, before we knew about DNA, we thought that we passed our proteins down to our offspring, and that's why they looked at us or looked like us. But amino acids are the building block for proteins. And I should go back and mention that lipids are made of fatty acids and a glycerol, and sometimes we add some things to them to, of course, change the structure and function. But proteins are made of amino acids, and they can be enzymes. We're going to see protein molecules in the membrane, like receptors and other enzymes like G proteins. Proteins are going to be found in cell walls. Ribosomes are made of RNA and protein, and antibodies, as I mentioned, are huge proteins. And they do all kinds of things for us. So far we've talked about carbohydrates, the primary source of energy made of monosaccharides, lipids, a secondary source of energy made of fatty acids and glycerols, and proteins, which are made of amino acids and have many functions. Last, we'll end talking about the nucleic acids. And you have two examples of nucleic acids, DNA and RNA. The Na stands for nucleic acid. DNA, the one most people have heard of, is named after the sugar that is in this monomer of this huge molecule. So DNA is made of these things called nucleotides. And nucleotides are made of sugar phosphates and nitrogenous bases. So it's named after the sugar, which is called deoxyribose. See the ose suffix? That's how you know it's a sugar, right? We just learned that. So DNA, deoxyribonucleic acid, is a nucleic acid made of nucleotides with the sugar deoxyribose in it. Ribonucleic acid is also a nucleic acid made of nucleotides, but the sugar in the nucleotide this time is not deoxyribose, it is ribose. Now, what is the function of DNA? It makes up your genes found in your chromosomes, and this determines inheritance. This is what we pass down to our offspring. Where RNA is going to be important for using those genes to make proteins. That is the job of many RNAs. So as I mentioned, macromolecules are these large molecules, and they are made from little smaller molecules, and we've already kind of gone over these, so some of this is going to be review. Polymerization, so a monomer, mer means part, mono means one, so monomer is one part, polymer is many parts. So polymerization is the building of a polymer from many monomers. So macromolecules, as we mentioned, can be structural or be molecular messengers like our antibodies. They can be energy sources and enzymes, even nutrient stores. We store up the energy, and sources of genetic information. So carbohydrates, a saccharide means sugar. So a monosaccharide, as I said, was a simple sugar. And this can have as low as 3 carbons with hydrogens or 7 carbons with hydrogens. Remember, they're organic molecules. Everything we're talking about is organic molecules, so they're going to have carbon and hydrogen with them. A disaccharide is going to be 2 monosaccharides held together, and a polysaccharide is going to be usually 5 or more of these monosaccharides held together. And those monosaccharides and disaccharides and a few polysaccharides do end in that suffix ose. Now, generally speaking, if you talk about hexose, hex means 6 and pent means 5. So that's a 5 carbon sugar hexose, and hexose is a 6 carbon sugar, and pentose is a 5 carbon sugar. So glucose, here is glucose. Let's count the number of carbons. 1, 2, 3, 4, 5, 6. Is it a pentose or a hexose? It is a hexose, and it can be in this linear form or in a ring. Galactose 1, 2, 3, 4, 5, 6, another hexose. And so it can also be linear form or a ring. Fructose, it's another hexose. And all of these are monosaccharides and hexoses. Now, you get when you're teaching this and when you're learning it, you get really tired of drawing these out. All these little carbons and hydrogens and oxygens. By the way, if you'll notice, these carbons are represented by the corners in this structure. So this didn't make a corner, it made a O, so that's an oxygen there. But here, let's count these carbons. 1, 2, 3, 4, 5, and then here's the 6th carbon like a flag on top. This is an easier way to draw this exact same thing. So where are the carbons? 1, 2, 3, 4, 5, and this will be a 6 carbon molecule because of these lines. And then when you add these together, you have a disaccharide. So monosaccharide, disaccharide, and when you add many together, you end up with a polysaccharide. So these could be glucose, lactose, fructose. This could be lactose, sucrose, or maltose. This could be a picture of glycogen, starch, things like that. So what holds these monosaccharides together is called a glycosidic bond, glyc also meaning sugar. So glycosidic bonds, your book defines it as carbons on adjacent sugars that are bonded to the same oxygen atom like links in a chain. And so you're seeing these two carbons coming together to form a glycosidic bond, this bond holding these two sugars together. Now, if you'll notice, if you look closely, the hydroxyl group over here and the hydrogen of this hydroxyl group on the adjacent sugar left as what molecule? Water. So it's losing water molecules when we're putting these monomers together to form these polymers. And so during polymerization, the process of making these huge molecules, they call that dehydration synthesis because we're forming water as a part of that chemical reaction. And so here's a bunch of polysaccharides. This is going to be what cellulose looks like. Cellulose, because of this structure, we are not able to digest very well. So this is fiber. Even cows, you've probably heard cows have 4 stomachs and they chew the grass. The grass is plants and their cell walls are made of the cellulose, and they have to spit it back up or throw up what they ate after it's mixed with enzymes in one of their stomachs, and they chew it, and we call that cud. They're working on breaking down this cellulose for energy. We are unable to do that, and I'm okay with that. I don't want to throw up and re-eat. Gross. Now, starch we can eat, and this is what starch looks like. And so we are able to break these little sugars off and get energy from that. So we saw cellulose, and it's pretty strong and rigid, and this is going to form the cell wall of algae and plants, and it is one of the most common substances on Earth. There's tons of plants and there's tons of algae in the oceans and lakes, and so we see a lot of cellulose. Remember, it is digestible only by bacteria, fungi, and protozoa, and bacteria is living in the cow's stomach, so that's what's helping the cow. I think about, for protozoa, termites have a protozoa living in them, and so they eat wood, which is pretty much pure cellulose, and that protozoa breaks the cellulose down into small sugars so that the termite can live. Agar, you want to star that word, write that word down, agar. It is a polysaccharide, and we get it from seaweed, and we're going to use agar a lot in microbiology to create culture media to grow these microorganisms on. So it is a polymer of galactose and sulfur-containing carbohydrates for these different micromolecules to use. Chitin, this first one, this is another polysaccharide, chitin. It is a polymer of glucosamine, and we see it in the walls or exoskeletons of fungi. Peptidoglycan, I always smile when I see this word because I had a former student call it peptidoglycan. But peptidoglycan is a polymer also. It's still a type of sugar, so we call it a polysaccharide. And it is going to be linked to little peptide fragments. And this one we want to star because it's going to be found in bacterial cell walls. And the lipopolysaccharides are a combination of lipids with polysaccharides, and these are going to be found in the outer membranes of gram-negative bacteria. So in microbiology, we are doing tests on bacteria to identify them, and we could use this information to know that their gram-negative bacteria have these lipopolysaccharides. And these chemicals, this lipopolysaccharide, is what is responsible for fever and shock when you have an infection with that bacteria. Glycocalyx is a polysaccharide that is bound to some proteins, and we see this for cells to attach to other cells, and we see this in receptors on membranes. Now, starch and glycogen, which I already mentioned, are polymers of glucose, and we store those in cells. And so starch is stored in plant cells and glycogen is stored in animal cells. And these have to be broken down by enzymes in the cell and used for energy. Now remember when we were making polymers from monomers, we called that dehydration synthesis. Dehydration, lose water, synthesis, create. Okay. Well, hydrolysis, hydro is water, and lyse means break. In hydrolysis, it's actually the opposite of dehydration synthesis. Instead of losing water, we're going to add water in this reaction. And instead of building up a complex molecule, we're actually going to digest or break down this polysaccharide. So lyse means to break. So hydrolysis, use water to break things down. Now, lipids are unique because they are the only macromolecule, they are the only organic molecule, lipids, that are not soluble in polar solvents like water. So, not soluble won't mix with water, and you know this if you've ever mixed oil and vinegar. Vinegar does mix with water, and oil does not, and so you'll see this layering. But lipids are soluble in nonpolar solvents like benzene and chloroform and other lipids. So that's important to note. They are nonpolar. There's a rule, polar mixes with polar things and nonpolar mixes with nonpolar things. And lipids are the only macromolecules that are nonpolar. And these lipids are super long, and they're mainly made of these long hydrocarbon chains that are hydrophobic. Remember, fear of water. Lipids do not mix with water. So one type of lipid, a very common one, is fat, triglyceride. And this is fat and oil, and this is where we store energy. This is your second energy source. If you want to burn fat, I've got some good news and bad news. You can do it, but you have to stop eating carbohydrates. So you're going to have to sugar detox, which is tough, and then have a change of lifestyle where you don't eat a lot of sugar, and then you'll start burning these fats. So these triglycerides are made of one molecule of glycerol and 3 fatty acids, and that's why they're naming it a triglyceride. The tri means 3, and it's counting the 3 fatty acids. The fatty acids are the long chains of carbons and hydrogens. We'll see them in a second. Glycerol is a 3-carbon alcohol with 3 hydroxyl groups that serve as binding sites. So here's the glycerol. Here's those 3 hydroxyl groups that make this an alcohol. And then here are the long fatty acid chains. I told you they're mainly carbons and hydrogens. So lots of bonds, lots of energy in here. When we bind these two together, look, H2O, H2O. We're going to lose 3 water molecules. We are going to call that dehydration synthesis. And we form what's called ester bonds. And so this is your finished product of a triglyceride. You want to break this fat down, you're going to have to bring water back into the story and replace those hydrogens and oxygens, and you'll have a glycerol and 3 fatty acids. Fatty acids can be classified as saturated or unsaturated, and I've got a way for you to remember these, so write this down. This is a test question. Saturated fats have straight chains. Look how straight this chain looks. Even this is pretty straight. Okay, so straight chained. Single bonds only. Only single covalent bonds around these carbons, in between these carbons. So straight chains, single bonds only, saturated, solid at room temp. When you have oil or fat, really not oil, but when you have fat, it's solid at room temp. I think about grease or butter. It's got saturated fat in it. This is what can clog arteries if you eat too much. And notice these carbons are surrounded by hydrogens. Now, how did I just help you? Every term I used started with a S. Saturated, straight, single, surrounded, and solid. And so here is a saturated triglyceride. 3 fatty acids, all of them straight, bound to this glycerol. And so this is fat, like palmitic acid. The second type is unsaturated fatty acids. So they are not going to be straight. Notice the kink here, or the bend in the molecule. Everywhere, or almost everywhere that there's a double bond, it's going to off-skew the shape a little bit, and so it's not going to be straight. It's not going to just have single bonds. Sure, there's some single bonds in here, but there are now, what? Double covalent bonds. I see 1, 2, 3 double covalent bonds. So it is not straight, it is not single bond, and it's not surrounded by hydrogens. We have hydrogens not in these spaces. And it will not be solid at room temp. This will be liquid at room temp, like oils. So saturated fatty acids, all carbons in the fatty acid chain are single bonded to 2 other carbons and 2 hydrogens, where unsaturated fatty acids have at least 1 double bond between those carbon atoms. So triglycerides are one type of lipid. Here is our second type of lipid, a phospholipid. And in your homework, you're going to want to know the parts of your phospholipid. So phospholipids still are made of a glycerol. So here's the glycerol. But if you'll notice, we don't have the whole glycerol, and we don't have 3 fatty acids. We only have 2. Because we've removed a fatty acid and a piece of this, or fatty acid, the glycerol has changed its shape and is binding with a phosphate group. Remember, I told you that when we add functional groups, it totally changes the molecule. Well, we're adding this functional group, this phosphate group, right here. So it's taking the place of the third tail, or the third, what are these fatty acids? Ooh, notice this one's saturated and this one is unsaturated. So they don't all have to be saturated. They don't all have to be unsaturated, okay? So what happens, because we've added a phosphate group up here to this glycerol, we've made this molecule change structure and change its behavior. And this area, this region up here, this phosphate head, is going to be charged now with a negative charge, and therefore is polar. Now, these fatty acids are still nonpolar. So what am I saying? Remember, nonpolar does not mix with water or anything that's polar. And polar things do mix with water. So the polar head is now hydrophilic, and the nonpolar tails are hydrophobic. And because of that, phospholipids will layer up. They can do this is the most common way. If you put a bunch of phospholipids in water, they can make a phospholipid bilayer. So 2 layers of phospholipids where the phosphates, which are polar and mix with water, are attracted to water. We call those hydrophilic. And the tails want to get away from water, so they're trying to get towards the other tails. Nonpolar likes to mix with other nonpolar things, and so they invert inwards. And this is what makes up biological membranes. And then we do have some lipids that will arrange this way in a single layer in water. So as I mentioned, membranes are made of 2 layers of phospholipids. And so the polar heads of the phospholipids are attracted to the water on both sides, and the nonpolar tails are repelled, so they're trying to stay inside. So the hydrophilic face of the membrane, this edge, orients itself towards the solution, where the hydrophobic tails make up the bulk inside of the bilayer. And this has a whole lot of purposes in biology. This allows this membrane to be selectively or semi-permeable and remain fluid. Now, what I needed you to learn in your previous biology classes is that cholesterol, which we'll talk about in a second, is a 4-carbon hydrogen ring structure. It's a lipid, and because it's a lipid, it can hang out with these fatty acids in here, and these cholesterols in the membrane help keep the membrane rigid but fluid. So as I mentioned, cholesterol is in the cell membrane and reinforces it. It's also in some bacteria called mycoplasmas. But cholesterol is a steroid, and a lot of your hormones that are steroids are made from cholesterol. And these are these ringed compounds that are found in cell membranes and animal hormones. Ergosterol is found in cell membranes of fungi, so kind of similar to cholesterol. So what is wax? Wax is these long chains of alcohol and fatty acids bound with an ester. And they're soft and pliable when they're warm. They're also water resistant when they're cold. So this function of wax is natural waterproofing of your skin. It doesn't keep water out of your skin, it keeps water in your skin. And so that's what it's doing for you. Wax also waterproofs fur and feathers. And bacteria that cause TB and leprosy, they produce a wax that actually repels some of the stains we'll use in lab. And that helps us identify it, but also damages the body of who they're infecting. So far, we've talked about carbohydrates. We built polysaccharides from monosaccharides. We've talked about lipids and the triglyceride, the phospholipid, the steroids and waxes. And now we're with proteins, the major complex molecule of the organic molecules. And this determines, like, how you build this determines the structure and then how it functions, and then what proteins the organism has in it determines structure and behavior and even unique qualities of individual organisms. So what are proteins made of? Carbs are made of monomers. Lipids are made of fatty acids and glycerols. Proteins are made of amino acids. And there are 22 naturally occurring amino acids in nature, and we link them together by something called peptide bonds. So here are the 22 amino acids and their abbreviations. I want to mention cysteine once again because of its sulfur molecule in there. Methionine is really popular. It's the first amino acid on all proteins. And then all these others have been named. So the basic structure of an amino acid. Amino acids are the basic structure of proteins, but what is the basic structure of an amino acid? Well, they're carbon-centered, so each of these is going to have a carbon at the center with an amino group on the side, so that's the nitrogen with the two hydrogens, so that's why it's called an amino group, or amino acid. It's amino, and this is the carboxyl group, which forms acids. So that's amino acid, amino acid, amino. All of these are what? Amino acids. So what's different is their R group. That's the unique, what makes one amino acid unique from another. And so you can put a bunch of different things up here. Remember, cysteine has that sulfur I told you right here. Phenylalanine has a carbon ring. So does tyrosine. Valine has carbon branches. Alanine is super simple. And so all of these different amino acids are different because of their different R groups. A peptide is a molecule composed of short chains of amino acids, and they named it a peptide because the bond between amino acids are called peptide bonds. A polypeptide has more than 20 amino acids. So not all polypeptides are large enough to be considered proteins, according to this book. But generally speaking, most polypeptides are going to be proteins. Now, proteins have specific shapes, and you do want to, everything I'm reviewing and saying pay attention to is test material. And protein structure is also test material. And there are 4 levels of structure, and you start with the primary, the first structure, and that's just the basic building block of this protein you're making. So the foundation is which amino acids do you have, how many of them, and what order are you going to put them in, because they're going to be in a long string. So that's the primary structure. It's formed by, do you remember the name of the bonds between amino acids? Peptide bonds. So the bond here in the primary structure is peptide bonds. In the secondary structure, these functional groups and your amino groups and carboxyl groups start interacting with each other, and they make 2 different shapes. An alpha helix, which is like a helix. Think of like the double helix is 2 strings kind of ribboned, while alpha helix is 1. And beta pleated sheets are like an accordion. If you fold the paper up and like an accordion, it'll go up and down, up and down, up and down. The tertiary structure is when the R groups start interacting. So those 2 cysteines bind to form disulfide bridges, or other, if it's a polar R group and another polar R group, they're going to be attracted to water. And so they're going to form these funky shapes, okay, a folded polypeptide. Every protein has to have these 3 levels. First, you got to make the dang thing out of amino acids, put them in an order, and bond them together by peptide bonds. Then the functional groups on the sides are going to form these beta pleated sheets or alpha helices. Sometimes you'll have just a random coil. And then when the R groups interact, it's like taking a bunch of these and folding them up on each other. Some proteins will have quaternary structures because quaternary structure is just multiple tertiaries put together. So notice the one we just built, it's a part of this larger protein. So a quaternary structure has 2 or more of these polypeptide chains. So in our primary structure, we have a polypeptide chain with a specific order bound by peptide bonds. in our second level of protein shaping, we have the beta pleated sheets and alpha helices. Then the R groups interact and form, oh, by the way, these are going to be hydrogen bonds holding these together in the secondary level. In the tertiary structure of a protein, you can have hydrogen bonds. It's whatever the R groups can form between each other. It can be polar interactions and nonpolar interactions. It can be covalent bonds. It can be disulfide bridges, ionic bonds, van der Waals forces, hydrogen bonding, everything to make this a specific shape. And only proteins that are made of more than one of these has what we call quaternary structure, and it's their R groups interacting with R groups on a different polypeptide chain. So again, all those types of bonds are used to hold it together. So the function of proteins, a popular one is enzymes, and enzymes speed up chemical reactions in cells. And they are very specific to their substrate because of this shape and these levels of folding. By the way, if you change this structure, if I take tryptophan and move it over here and move valine, and I just put this, I use the same amino acids, I just put them in a different order, these shapes will change, these shapes will change, and these shapes will change. And so that's why this is the primary level, because it determines how these shapes are formed. And these. And these. So we can just change one or two things and have a totally different shaped protein, which allows us to have thousands upon thousands of different shapes, and how enzymes can have unique patterns and their binding sites binding for specific substrates. As we've mentioned, antibodies are complex glycoproteins, and they are going to have specific attachment regions where they're looking for bacteria or viruses or other microorganisms, part of your immune response. The native state, that word native, that's going to mean the functional three-dimensional form of a protein. So you haven't screwed it up at all. Where denature is when you change the shape and now it no longer functions. So when you denature a protein, you're going to do that with heat or very strong acids, even strong bases, some alcohols, disinfectants, things like that. You are disrupting those chains, those bonds within the protein, and now they are no longer functional. So if you have an enzyme and you heat it up, that is going to change the shape of the enzyme and it will no longer function, and we'll say that it's been denatured. But if the enzyme is functioning, it's in its native state. Last, we're going to talk about nucleic acids, the NAs in the DNA and the RNA. Now, DNA does have genes inside them. They're special coded nucleotides that have instructions for making all the proteins of the cell. Where RNA are helpers in taking the message from DNA, carrying that to a ribosome, and actually creating the protein. If you think about DNA, it's like a recipe and it stays in the kitchen. But RNA is like if you copied down that recipe and you took it to a friend's house and you made the product, and that would be the protein. So the original recipe that has to stay in my kitchen is DNA. The copy that you made is RNA. The ribosome is like your kitchen. And then the meal is the protein that the DNA had instructions for. So DNA and RNA are both made of repeating nucleotides, and nucleotides are made of a sugar, a phosphate, and a nitrogenous base. The sugar is what determines the name of the structure you're building. So if you're using deoxyribose, it's DNA, and if you're using ribose sugar, it's RNA. And these are 1, 2, 3, 4, 5 carbon sugars. So they are pentoses. We've seen hexoses, now we see pentoses. And so deoxyribose is a sugar, and ribose is a sugar with 5 carbons. So nucleotides, which make up DNA and RNA, are made of sugars, phosphates, and so The phosphates you can't see here, but they are going to be bound to these sugars. The blue ribbon here is sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then also a nitrogenous base. So the nitrogenous base is kind of the language of DNA and RNA. And both of these have 4 nitrogenous bases, 4 types. DNA has adenine, which binds to thymine, cytosine, which attracts to guanine. Okay, one more time. DNA is made of nucleotides, which are made of the sugar deoxyribose, phosphate groups, and adenines, which bond to thymine, and cytosines, which bond to guanine. ATCG. Ribonucleic acid is made of the sugar ribose in its nucleotide. It still has phosphates, the same exact kind as DNA, but and it's also going to have nitrogenous bases, but it does not have thymine. Instead, it has uracil. So what binds with adenine in DNA? Thymine. What binds with adenine in RNA? Uracil. And guanine and cytosine are still there and still are attracted to one another and bond to one another. We can classify these nitrogenous bases as purines and pyrimidines. Purines, there's this famous saying, it's called pure as gold. Your adenines and your guanines are your purines. Okay? Adenines and guanines are purines. This picture is just showing you the difference between deoxyribose and ribose. We have already looked at that. Purines are adenine and guanine. How are you going to remember that? Pure as gold. Pyrimidines look a little different. They're just one ring. Pyrimidines are going to be the rest of them. So not adenine, not guanine. So what do we have? Thymine, cytosine, and uracil. Adenine, what's cool is these purines, these double ring structures, are attracted to a single ring structure. So who is adenine attracted to? Well, in DNA it's thymine, but in RNA it's uracil. And who is guanine always attracted to, no matter if you're DNA or an RNA? It's cytosine. And we call those base pair rules. So DNA contains all nitrogen bases except uracil. And the nitrogen bases are covalently bonded to deoxyribose, where RNA has all the bases except thymine. And they are covalently bonded to ribose. The sugars are different. And we are going to pause right there.